Good morning. Thank you very, very much, Julie, for joining us today on this really special occasion where Mahmoud and I have the honor to spend a little time with you and learn about all the things that have made your career so illustrious and so impactful in the biomedical sciences. I'd like to start with some of your background. What were the motivating factors as a child that led you to eventually pursue more serious studies in college and subsequently?
When I think about it, what really was important was two components. First, there is, I think, this connection with nature and how we spent a lot of time in my childhood, both exploring nature, hiking, and a lot of it was even just that we were in Colorado in the summers, and the kids could just go out, out from our apartment and go up into the hills and the mountains. And there was a sort of modestly supervised component of just looking for things and trying to see what was on the landscape, and having that be part of how we played together as children. So that's one part of it. And the other part of it, I have to say, is these books that my parents would give us. And I mean, reading books, of course, but also just these puzzle books that we used as kids. And so my favorite puzzles were always the ones about, you have four people and they live in four houses and they have four different cars, and their houses are painted four different colors. And you have to figure out from the clues if they say, like, Mr. Green does not live next door to the man with the brown car. And you would have to figure out then who lives in what house. And I came to see that I really enjoyed doing these kinds of puzzles where you have data, and from the observed data, you have to then come up with a model of how would you explain what is the simplest explanation for how this data is all compiled into a data set. And so I think that part of what we're doing in these careers is to understand, how do our brains work? And so when I think about that, it's for me that I enjoy organizing large data sets and thinking about what are the patterns, and that it could have been that I would have enjoyed being a crystallographer, where you get sort of data and to figure out what is the molecule. But it also could be something like microbiome science, where you get all of these, reads, and you have to try to assemble them into genomes, and you have to think about, how would this community assemble itself, and how could I learn that from 5 different data sets? There were several teachers who really impacted my career before I even entered sort of formal academia. And they all were important in the context of both providing confidence, but also helping to, steer a career. I do want to mention in particular my grandfather. I think the role of grandparents is really remarkable in our society, and anyone who has a grandparent involved in their life, it's a real blessing. My grandfather was a code breaker in World War II. And my sister and I used to write little codes and then hand them to him, and he could read them immediately. And it was a fascinating discovery for me that, that could be something of sort of reading codes that would be both part of how you served our country, but also highly developed as both something we enjoyed as kids, but also something that could be pursued as a career.
So now I completely understand why you got an undergraduate degree in mathematics at Amherst. You then went on to graduate studies at the Massachusetts Institute of Technology. After that, you decided to go into the code-breaking discipline of genetics and genomics. How did the interactions at MIT have an impact upon you? You probably interacted with some of the major players in molecular biology, and you developed your own way of approaching life and science.
It was a bit of a stretch to be a math major in college and apply to a PhD in biology. And I did have to write several letters accompanying my application saying, it is clear that I have not taken very many biology classes, but I have a deep interest. And perhaps it was, well, I was just fortunate that people could see how you could bring math into biology. I now know many programs have more of an applied math towards data science. But while at MIT, we built the first genetic map of the mouse and the first physical map of the mouse genome. And it was these same kinds of puzzles where we, one of the most important papers had been written that was really how to calculate a log score, the log of odds ratio score, where you would think about if I see this inheritance patterns of two genetic markers in an F2 cross, how likely would it be that I would just see that by chance versus that I would see that because these two markers were on the same chromosome and in linkage with each other. So I definitely was seeing how having the ability to state something as a formal mathematical hypothesis would be something that would help to move forward the field of biology. And it was very exciting for me to find people and have conversations where the training of how to go to the board and do a mathematical proof would be really coupled and embraced by people who were thinking about creating large data sets and having that kind of data to analyze it. It was not always easy in that at the original time, the Human Genome Project was just being funded, and I think there was still some trepidation of, was this really going to contribute to science? And was it going to be something that there was a huge investment in, but it didn't serve very many people? And was it really just highly technical work, or was there an intellectual component of it? So I think at the beginning of the Human Genome Project, we tried to construct those arguments about how understanding the information about a genome, how we were going to make that information publicly accessible, and how it would be used for many different biological questions and experiments, and also how what we were trying to do as genomics geneticists was something that perhaps required some slightly different training, but would be something that we would be bringing into biology, and it would be fully integrated in with biology. And now you see it even being part of the training program of graduate biology, that it was about working at intersections of fields.
I can tell you, listening to you, it's amazing. You know, it took me back to when I was a little boy in Lebanon and Beirut. And that's exactly what we did. I mean, we went outside, and my mom used to say, as long as you come back before sunset, you are okay. It was very safe. And really what's so critical when you were talking is I'm now looking at the present generation, the young. I'm a granddad, and I see the kids, what they do these days, is spending a lot of the time. Unfortunately, the world changed from when we were young. That's amazing what you said. And I really would like your thought, you know, what do you think our new generation, these kids will have to substitute for those going out and playing as much as we did?
I think they will still have it. We would do trips like that. You're right. We still live along the Potomac River, and there's a lot of paths around our area. I mean, my kids still go backcountry camping just with their friends and their backpacks, and discovering our country. So I hope it's still part of how people grow up. I mean, you're right. I lived during the school year in the inner city, and we had a lot of freedom, but it wasn't that we had to go other places to be able to just sort of go out into the mountains.
So, Julie, looking at your career, I remember the first time I met you at the National Institutes of Health. You were very deeply involved in trying to understand the skin microbiome. And since then, you've gone on to other very important nosocomial transmission events. But as a graduate student, it seems like you were mostly involved in transcriptional regulation, trying to understand how those sorts of factors impact biological expression. How did you evolve into studies involving the skin microbiome as a complex community? How do you think your findings have significantly impacted health and human diseases?
My graduate work was what we called at the time positional cloning, integrating the genetic and the physical maps of the mouse genomes in order to identify the underlying causative gene that was mutated in nude mice and rats. And so I had gotten some work with mouse skin mutants, and had decided at the end of my PhD that the Genome Project, it was clear it was going to go on and sort of really institutionalize the sequencing of the BACs and we were switching from YACs to BACs. It was institutionalizing sort of sequencing of the human genome. And I thought, well, I can take a couple of year gap from the Genome Project and come back to it. I understand the basis of how we are doing these experiments. And now is really the time for me to understand a biological system so that I can use genomic tools to ask questions. And the question I was most interested in was, how do you balance proliferation and differentiation and sort of homeostatic balance? And the question, it seemed to me, was being asked most profoundly in hematopoiesis, although there were other systems. But I am typically a person who sees, like, here's where there's a lot of emphasis, and maybe what's a little bit around the edges. Because if you go into a field that's a little bit around the edges, then you have some time in which you can sit and think about what are the questions you want to do. And you don't have to be running all the time, worried that someone else is doing the same experiment. That at least has been sometimes my philosophy. So I saw people asking that question in hematopoiesis, but I also saw that there are other systems in humans that also maintain stem cells into adulthood, including the human skin. And so that seemed to me that we could ask the same types of questions, but we would have more opportunities with the cell culture models and just the availability of human skin. I moved to go to what was and still is one of the very best labs in skin biology, Elaine Fuchs, to understand sort of a general concept of what are all the different ways in which we address questions in skin biology. And while I was in Elaine's lab, I was working on a transcription factor. KLF1 was controlling the switch of globin genes. And I thought if I found the cripple-like factor that was in the skin, I thought I had a good chance of finding the gene that was going to regulate keratin gene switching as part of the terminal differentiation process. Instead, it turns out KLF4 controlled skin barrier, which is part of the terminal differentiation process. And I started working then on KLF4 and also GATA3, which is another transcription factor used in hematopoiesis as well as in skin differentiation. So that seems like, yes, I had this sort of period of working intensely on transcription and thinking that I was going to bring genomics sort of to skin biology and use the skin system to answer questions about genomics. But then when I started looking at what genes were affected in the KLF4 knockout and in the GATA3 knockout, it was antimicrobial peptides that were being differentially expressed, which makes total sense if you start to think about, if you perturb the skin barrier, the skin needs to respond as a sort of a stress response. And it needs to think about, I need to rebuild the skin barrier as soon as possible. But in the immediate now time, I need to think about how am I going to control possible infections or pathogenic microbes. And I started seeing that KLF4 and GATA3 were upregulating different antimicrobial peptides. So it wasn't just that there was a sort of, go pattern. There was a tuning of it. And that started me thinking about, so how do we know what microbes are colonizing the mouse skin in these mutants, and how will that be affected in the different classes where KLF4 was more affecting the proteins and GATA3 was more affecting the lipids that were controlling skin barrier. And it's that kind of moment where I thought, I'd like to know what are the microbes, just at the time when we were really getting the ability to do 16S bacterial, marker gene sequencing. And it also was a fortunate time in my career in that I was just coming up for tenure, and it gave me a little space again to think about, what do I really want to do? And that the next step in the transcriptional factors was to make, targeted knock-ins and do chip, tagging and da-da-da. And I thought, well, I could do that. But here's this other thing that's brand new, microbiome sequencing. I could do that here at this, I'm in a genome center. I could do this here. And I just sort of shifted where things that I was doing with these transcriptional knockouts, I'll come back to them. I will definitely come back to them. The idea of host-microbe interactions is still what is really of great interest to me. But I did then go on probably about a 15-year detour of sequencing microbes still in the context of skin and understanding. It's been very important to me to work closely with dermatologists who structure the clinical studies, who make the decisions about what kinds of patients we're going to recruit, what types of evaluations we'll do, how should we, even how could we obtain the skin sample? Should it be a swab, a scrape, a biopsy? Always doing these experiments as a team.
I am really so glad you took this 15-year tour, okay? Because it enriched us with respect to the skin microbiome. And, it was fascinating to me when I read your paper about the topography of the skin microbiome and the lovely colors you came out. Different parts have different, distribution based on the niche where you are looking, whether it's moist or it's dry and this sort of thing. So that really was fascinating to me. And I think it established the way, like now when I start looking at the skin, being part of dermatology, we follow a lot of what you did. So that is really fantastic. What I want to ask you, however, at that time, we didn't know about Candida auris. Do you think if they were there, but we did not look at them? Or are you thinking of looking again on the skin to see, because Candida auris lives in the skin, so to see whether it is present in normal people or in patients? What do you think is the best way to know now, taking into consideration that this could be a nodus of infection for people?
Candida auris as an emerging pathogen was first described in a Japanese patient inside the ear, which is why it has the title Candida auris, same way as we talk about retroauricular crease behind the ear. But it was independently discovered, and now we use clade names, clade 1, 2, 3, 4, but those 4 clades are tens of thousands of SNPs apart from each other, but we have not found how a pathogen simultaneously emerges on 4 different continents and becomes something that has the ability to colonize humans and cause disease. So the CDC then went back, working with other countries, to go back and look at all of the banks of isolates that they had. And they were not able to, I mean, they did discover some isolates, I believe, from like 2007, 2008, but nothing prior to that. It isn't that it was infecting humans for decades and we missed it. The system of identifying an emerging pathogen was very successful, and it was picked up really pretty quickly as soon as there was a cluster of cases. And so then we thought, well, let's try to use genomics. And we have all these data sets that have been published, and maybe, as you're saying, maybe someone just didn't know to look for Candida auris. And there were a number of people, colleagues of mine at NCBI, colleagues of mine in the Genome Institute, working with the mycology branch at CDC, who were trying to screen all of SRA to find Candida auris. And we do not find it in earlier data sets. There has been work now in India by some of our highly esteemed colleagues to find Candida auris in the environment of both, the built environment and the foods, and also the natural environment. It probably still has the question of whether humans have contaminated those environments, or is that a reservoir? We are seeing at this point that there's a lot of human-to-human transmission, but certainly Candida auris and its ability to persist in the environment makes it quite a formidable pathogen. I work at the NIH. I was down visiting CDC. We're both part of Health and Human Services. And I had done some work on just characterizing the normal skin flora. And for the first time, had a chance to meet with the mycology branch at the CDC, Annalyn Seva, Snigdha Valavana, Tom Chiller, Brendan Jackson. And they said, we've got this, new emerging pathogen, would you take a look at it? And so we worked with the CDC on the original sets of isolates from the nursing homes in New York and the nursing homes in Illinois. But it was really that introduction and all of the background that the CDC and their work that they had done to basically demonstrate how important Candida auris was. And we were really at that point trying to demonstrate that Candida auris was a skin colonizer, that this wasn't simply a gut colonizer like Candida albicans, that it wasn't a gut colonizer with fecal patina on the skin, that it really was a skin colonizer. And so then working with my colleague Mihalis Leonakis at NIAID, we really were trying to do experiments both in mice and in humans to demonstrate that Candida auris does colonize the skin as its primary site. um, uh, as well as in some cases, also colonizing the GI tract, but that it really is a skin colonizer. That to me is still the question that sort of drives our research in my lab right now is how does a microbe tune itself to this environment? How has Candida auris adapted to live on human skin, to utilize the nutrients of the skin as its food source and persist?
I really love that actually, because as you know, the interest now, because in the skin, is to have decolonization model. And I saw your paper with Mikhail about the mouse model for decolonization. And I must say, I learned a lot from that, because we try to use the guinea pig model usually for skin infection, but your model was really very, very helpful in trying to see, can we have a model for decolonization and then try to discover some new antifungals that can do that. So this is very, very interesting. One thing which you said, why did we see Candida auris? And Arturo Casadevall, of course, talking about the climates and this sort of thing. So I would love to hear what you think about that with respect to the climate as a change, as a really inducer of, let's say, Candida auris.
I would say talking to Arturo inspires us all. He and I were talking on a more, granular level, and he really pointed out to me the temperature fluctuations across the skin, because I've wondered why we see so much fungal diversity on the feet when on the core body it's much more Malassezia dominated. And Arturo was telling me that there are these temperature, I knew about the temperature fluctuations, but that the feet is perhaps the most extreme of the fluctuations. And we put little temperature sensors on, and we had, people volunteer to wear these little temperature sensors all over. And it's true. I mean, the feet have this really tremendous variation in temperature. So Arturo is someone who is very playful about sort of having, spent time in nature and understanding that those big concepts can have, an impact on the science we do, and to phrase it in those kinds of ways. So for so many of the work that we do where we're looking at hospital outbreaks, we're taking genomics and we're trying to wind back the tape of saying, how did this happen? What is the, using genomics to then try to say, what is the most recent common ancestor and where did these SNPs arise from? I think Arturo's work is very important in understanding that there are these global changes and that they're going to change the places that microbes can live, and also sort of other, other things, ticks and insects. And and they're going to change where these infectious agents can live. And with that, we're going to see alterations in, diseases that you think, we never saw this kind of tick-borne encephalitis in this region of the world. Well, now we might start seeing it when we have these changes in the temperature. Yeah.
From the skin, you went on to the hospital environment and you started tracking these multi-drug resistant gram-negative bacteria. And you did some very significant work in understanding the transmission of these pathogens in a microbial community, specifically looking at carbapenem-resistant Klebsiella pneumonia, and then delving deeper into Acinetobacter baumannii. Going from your previous background and the complexity of the skin microbiome to the complexity of the hospital, how did you and Dr. Palomore approach these really complex issues, and what do you think was most significant even today?
I was fortunate to be introduced to my colleague, Patrick Murray, who ran the clinical microbiology at the NIH Clinical Center. And he was helping me and really providing the background and the intellectual input on microbiology, which I hadn't studied in graduate school. And working together on the skin microbiome, we got along so well. And I think it is really important when you find good collaborators. He was interested to learn about genomics. I was interested to learn about microbiology. To ask these sort of open-ended questions. Is there anything else we could work on? And he said, well, we do have these issues with, pathogens, and we've only been able to study it with pulse field gels. Well, I had run pulse field gels, but I understood how much more resolution we could get with genomic sequencing. But the crucial component of it is finding disciplines where you have the same, in a sense, you have the same training and you have the same way of asking questions, because as I was describing to you about genetic maps and how we have to calculate what is the odds that this could have happened by chance versus that this happened because there is a connection of these two markers on the same chromosome. That is the same model by which Dr. Tara Palmer, who was our hospital epidemiologist, asks a question about, I have 2 patients in the hospital with Acinetobacter baumannii. What is the chance that they came in simultaneously or concomitantly at the same time period with the same microbe, maybe because it's increasing in frequency or they came from the same facility, versus that there was a transmission in our hospital? It's really the same question. And then I think about, okay, so this is how we use genomics or genetic sequencing to ask that question. How would we use that question? How would we use genetics, genomics to answer that kind of question? And so exactly that. I remember sitting in a small conference room with myself, Evan Snitkin, and Tara Palmer, and we had just gotten back the sequence of the Klebsiella pneumonia isolate from the first 5 subjects at the NIH Clinical Center. And we had sequenced this patient number 1 who had been in the hospital earlier, because we were planning to use patient number 1 as the out group. But in fact, instead, what we found was that patient 1 was very closely related to 2, 3, 4, and 5. And I said, Evan said to Tara, he said, we think that these are all a cluster. And he said, but I think patient 1 independently infected patient 2 and patient 4. And then we have something which we don't understand, because we think that patient 4 infected patient 3. And so the numbers don't add up. And Tara said, no, that's exactly what I think happened too, because she was looking at patient trace data and seeing who was in the ICU together and who could have possibly, transmitted to each other. And that you don't necessarily always, you number the patients based on when you identify them, but a very sick patient could present with an infection or have a higher microbial load sooner than a patient who you identify a few days later because their underlying condition is different. So when we had that moment where we had 2 different types of information, but we had arrived at the same conclusion, I think that's when we all thought, oh, this is actually quite profound, that 2 very different methods from our 2 different disciplines would come to the same basic conclusion. And from there, we literally just sprinted for the next few months to figure out what else we could learn in this case.
All of us are not only investigators and researchers, but also mentors for young and upcoming scientists. As a woman who has accomplished becoming a member of the National Academy of Sciences and having all those tremendous honors, what advice would you give our young faculty or young investigators at the start of their career? What do you feel the most important things that define us as good scientists and as good citizens?
There's such a complexity that goes into our careers. For me, I've tried to say I like going into new areas of research, and I don't mind feeling uncomfortable in a situation. Like going into the skin microbiome, I could say, I know about genomics, I know about skin, I do not know about microbiology. And I don't, and I can't let that stop me. I just find people who can help me learn about microbiology and read papers, but I am comfortable being uncomfortable. And but then, I balance that by saying that I also really value my colleagues and friends, and as you said, also to be a good citizen, to think about what are the projects that I'm working on, and to think in advance about the translational arc. For me, working with clinicians means that before, when I was working as a more of a basic scientist, I would hope that someone would pick up these findings I had and develop them. But it's been more rewarding for me to work with clinicians from the beginning, so that translational component is built into the project. And I'm thinking more myself now about how even to include a communication and a public policy component of the work that we do, so that we engage in those areas also as citizens, and don't just think, oh, someone else will pick this up and try to communicate the findings of this work. But in terms of young scientists, I know they have a tremendous amount of pressure and obligations, and the last 2.5 years has just been so hard, especially for early career researchers. So I guess I think where I'm putting my time now is that as a really more of a mid-career scientist or scientist, I'm really trying to invest a lot in community building, and holding meetings and thinking about how we can just have communication of ideas and work together on projects and just maintain and build scientific community, because I do believe that the community is stronger than each of us as individuals.
On behalf of Mahmoud, Pathogens and Immunity, and myself and our editorial board and our readership, we really thank you for your time today, for your insights, and for sharing all your formative experiences and for your vision of the future. Our society has been significantly challenged these past 3 years, and it is so important to see successful women scientists like you keeping us going forward. Your leadership and your contribution to the field are very, very meaningful to all of us. Please note that Julie will be presenting Tracking an Emerging Pathogen Candida auris in Nursing Homes with Genomic-Epidemiology at the upcoming Keystone Symposia, the Human Microbiome, Ecology and Evolution. This program will take place December 4th to 7th in Fairmont Banff Springs, Alberta, Canada.